Welcome. We're discussing a fascinating field today, quantum biology. Our focus is a comprehensive review titled Quantum Biology, an Update and Perspective, published in Quantum Reports in 2021 by a large team including Youngchan Kim, Federico Bertagna, John Joe McFadden, and Jim Al-Khalili, among many others. Rachel, to start, the very idea of quantum mechanics playing a role in biology seems counterintuitive to some. Can you explain what quantum biology is and why it is attracting so much attention?
That's a great starting point, Jake. Traditionally, biology has been seen as operating in the classical world, while quantum mechanics was reserved for physics and chemistry at a much smaller scale. However, living systems are fundamentally quantum mechanical at their core. Their molecular and atomic machinery, like everything else, follows quantum laws. The crucial distinction in quantum biology is between what we call trivial and non-trivial quantum effects. The paper notes that early pioneers like Erwin Schrödinger, with his book What is Life, first argued affirmatively that quantum mechanics plays a fundamental physiological role.
So what's the difference there? What constitutes a trivial quantum effect versus a non-trivial one in a biological context?
Well, trivial quantum mechanics is everywhere. The structure of electronic orbitals, chemical bonds, these are all governed by quantum laws, but they are so fundamental that they form the bedrock of chemistry and structural biology. We do not consider structural biology a branch of quantum biology for this reason. Non-trivial quantum effects, on the other hand, involve phenomena normally confined to the quantum realm, like coherence, tunneling, entanglement, or spin. These effects are thought to be highly improbable within the warm, wet, and disordered environments inside living cells, because they typically cancel out at the macroscopic level due to decoherence. For an effect to be non-trivial, it must explain or drive a biological process that evolution has likely optimized. Biology must have evolved to exploit it.
I see. So it's not just that life is made of atoms, which are quantum, but that life might actively exploit these more exotic quantum phenomena for a biological advantage. The paper highlights several areas where this might be happening. Let's delve into enzyme-catalyzed reactions first, specifically hydrogen transfer.
Exactly. Enzymes are proteins, often described as the molecular workhorses of cells, and they accelerate chemical reactions far more efficiently than inorganic catalysts. Among the most important reactions they catalyze are hydrogen transfer, H-transfer reactions, including proton, H-atom, and hydride transfers. The debate about nuclear quantum tunneling in these reactions has gone on for decades, but it's now well established that H-transfer reactions, enzymatic or uncatalyzed, involve some degree of H-tunneling. This is due to the small mass and inherent positional uncertainty of the proton.
So protons are light enough that they can actually tunnel through an energy barrier rather than having to climb over it, which is a purely quantum mechanical effect. How do we measure this tunneling in enzymes?
The primary kinetic isotope effect, or KIE, is a reasonable first approximation. It is the ratio of rate constants for transferring a lighter isotope versus a heavier one like hydrogen versus deuterium. Because of the mass difference, they have significantly different tunneling probabilities. If an enzyme's KIE is much larger than the theoretical semi-classical limit, which is around 7 at room temperature, it strongly suggests H-tunneling is a predominant reaction pathway. We must also remember that the degree of tunneling is very sensitive to the barrier height.
That's a clear indicator then. The paper also mentions the importance of protein dynamics. How does the enzyme itself, meaning its movement and structure, facilitate this quantum tunneling?
Well, enzyme catalysis primarily works by lowering the barrier height. Protein dynamics also play a role. The specific configuration where tunneling is most likely to occur is called the tunneling-ready configuration, or TRC. This configuration is achieved through classical reorganization, and then rapid small-scale protein vibrations can further compress the donor-acceptor distance, enhancing the tunneling probability. These are often referred to as promoting vibrations. The temperature dependence of the KIE can even be modeled using vibronic models that directly relate tunneling probability to this tunneling distance.
So the enzyme is not just a static scaffold. Its movements are actually part of the quantum process. The paper points to light-activated enzymes as excellent model systems for studying this. Can you elaborate on that?
Well, many enzyme reactions are thermally activated, making them hard to study over wide temperature ranges and very short timescales due to limiting factors like substrate binding. Light-activated enzymes overcome this. You can trigger catalysis with a single pulse of light, allowing for studies from femtoseconds to seconds and across various temperatures. Protochlorophyllide oxidoreductase, or POR, is a prime example. It's a chlorophyll biosynthesis enzyme that catalyzes sequential hydride and proton transfers. Studies on POR have shown that both hydride transfer reactions proceed by tunneling and are coupled to promoting vibrations. Its unique properties, like separate H transfer steps and a consistent temperature-dependent KIE across species, make it a powerful system for understanding how enzymes evolved to exploit quantum tunneling.
That's a compelling case. Moving on, photosynthesis is another biological process often cited for its quantum aspects. What's the latest thinking there?
Photosynthesis is remarkably efficient at its initial steps of light harvesting and energy transport, achieving near unity quantum efficiency despite operating in warm, noisy environments. This efficiency has led researchers to propose that non-trivial quantum mechanical effects are at play. Initially, energy transfer was explained by Forster resonance energy transfer, or FRET, which describes incoherent hops. But it became clear that FRET alone couldn't explain the process, especially with strongly coupled chromophores, where excitation is delocalized, forming what are called Frankel excitons. Theoretical advances, like using the hierarchical equations of motion or pseudo-mode methods, help model these intermediate coupling regimes.
And this is where quantum coherence comes in, right? The idea that energy can explore multiple pathways simultaneously, leading to a quantum walk of energy transfer.
Precisely. The development of techniques like two-dimensional electronic spectroscopy, or 2DES, has been crucial. In 2007, Engel et al. Showed oscillating cross-peaks in the Fenner-Matthews-Olsen complex, which they attributed to electronic coherences persisting for hundreds of femtoseconds, even at low temperatures. While controversial, this suggested that quantum coherence might enable the system to efficiently find the best pathway to the reaction center. The key question has been whether these long-lived coherences observed in the lab are relevant in noisy biological environments.
That's the big challenge, isn't it? How can such delicate quantum effects survive in the warm, wet, and noisy cellular environment?
Indeed, but a key concept emerging is that the environment might actually enhance coherence, not just destroy it. Experiments on the Fenner-Matthews-Olsen complex at physiological temperatures demonstrated quantum beats lasting for 300 femtoseconds. Theoretical studies predicted that intermediate noise levels, like those at room temperature, can enhance energy transport when the dephasing frequency is tuned to the excitonic energy gaps, a phenomenon sometimes called environment-assisted quantum interference. This suggests a more complex interplay between quantum and classical mechanics.
It seems biology might have found a way to use noise to its advantage. Let's shift to another fascinating area: magnetic field effects on spin-dependent reactions, particularly magnetoreception. Birds using the Earth's magnetic field to navigate, for instance.
That's right. The most prominent example is the radical pair, RP model, of animal magnetoreception. Radicals are molecules with unpaired electron spins. When two radicals are generated simultaneously, their electron spins are initially correlated in either a singlet or triplet state, which have different reactivities. If this interconversion between spin states is influenced by a magnetic field, then the outcome of the reaction can change. This influence comes from local magnetic interactions of nuclear spins within the radicals, known as hyperfine couplings, HFCs.
This sounds like a huge challenge to thermodynamics, though. The Earth's magnetic field is incredibly weak. How can such a tiny force possibly influence biochemical reactions, given the thermal energy inside a living organism?
That's a critical point. The interaction energies are orders of magnitude lower than thermal energy. However, the radical pair is, at least transiently, far from thermodynamic equilibrium, often due to how it's generated, reflecting the Pauli exclusion principle. The spin dynamics are not bound by the KBT argument until the system relaxes to a Boltzmann distribution. This gives the weak magnetic field a window of opportunity to alter the population of spin states and thus the outcome of the reaction, provided the spin correlation persists long enough, usually around 1 microsecond, to be sensitive to the geomagnetic field.
So it is not about changing the energy of the reaction directly, but rather influencing the probability of certain spin states during a fleeting non-equilibrium phase. This allows a weak magnetic field to have a disproportionate effect. What are some of the candidates for these radical pairs in biology?
Flavin-dependent photoreceptors like cryptochrome are key candidates believed to be responsible for magnetoreception in birds. Electron transfer in these molecules can generate radical pairs sensitive to light and magnetic fields. Other examples include light, oxygen, or voltage sensing domains, and even light-driven enzymes like protochlorophyllidie reductase, which we discussed. The paper also mentions that spin correlation can arise from bond homolysis, like in vitamin B12 biochemistry, and even from random encounters of freely diffusing radicals, known as F pairs, which are implicated in the magnetic sensitivity of lipid peroxidation.
It's a complex picture, and many conditions need to align for these effects to manifest. Now let's explore proton tunneling in DNA. Mutations, the driving force of evolution, are often seen as random errors. Could quantum mechanics play a role here?
Absolutely. Watson and Crick themselves, in their 1953 paper, proposed a subtle mechanism for spontaneous mutations based on tautomeric forms of DNA bases. Each base can exist in different forms, and if one base adopts its rare tautomeric form, like A-amino T enol or G-enol C-amino, it can lead to mismatches like AC or GT. Crucially, the geometry of these tautomeric mismatches can mimic the canonical base pairs, allowing them to evade fidelity checkpoints during DNA replication.
So the wrong base could temporarily assume the right shape, leading to an error in replication. How does quantum tunneling fit into this tautomerization?
In 1963, Per Olav Lodin proposed that quantum tunneling could facilitate the transition to these energetically less favorable tautomeric forms. He suggested that the canonical AT and GC base pairs could convert via a double proton tunneling process across their hydrogen bonds. If the DNA strands separate while in these tautomeric forms, they could then lead to mutations in the next replication round.
That's a direct quantum mechanical mechanism for mutation. Is there experimental evidence for this?
Recent studies using NMR relaxation dispersion have provided strong indications. For a GT mismatch, researchers observed a kinetic isotope effect, where the exchange rate between wobble and Watson-Crick-like forms was slower in heavy water /D2O than in normal water /H2O. This suggests a proton transfer in the transition state. However, to definitively attribute it to quantum tunneling, further experiments measuring its temperature dependence are needed, similar to what we discussed for enzymes.
So still a lot to uncover, but the theoretical framework and some experimental hints are there. Next, let's talk about fluorescent proteins. How do these common biological tools fit into quantum biology?
Fluorescent proteins like GFP are widely used in cell biology. They possess anomalous photophysical properties that challenge classical explanations. For instance, excitation energy transfer between fluorescent proteins was thought to be incoherent, limited by distance and rapid vibrational decoherence. But recent experimental evidence suggests otherwise. The beta-baryl structure, for example, isolates the fluorophore, making it surprisingly resistant to external quenchers.
What kind of evidence? What's happening at the quantum level with these proteins?
Studies have shown surprisingly long dephasing times for vibronic wave packets in GFP, much slower than organic fluorophores. Also, fast anisotropy decay constants in YFP dimers indicate rapid bidirectional energy flow consistent with coherent energy transfer. Even more striking, in 2017, Shi et al. Demonstrated the generation of polarization-entangled photonic states from enhanced GFP molecules at room temperature. This indicates EGFP fluorophores are remarkably insensitive to environmental decoherence, likely due to the isolation provided by their beta-baryl structure.
That's quite remarkable, maintaining entanglement at physiological temperatures. And what about excitonic coupling?
In 2019, Kim et al. Found further evidence of room temperature coherent molecular interactions in dimeric yellow fluorescent protein Venus A206. They observed ultra-fast energy transfer and Davidov splitting in circular dichroism spectra, which is a quantum mechanical manifestation of excitonic coupling. Their photon anti-bunching experiments showed that dimeric Venus A206 physics behaves as a single quantum entity, not just two independent emitters. This strongly supports the idea that FPs have evolved mechanisms to prolong quantum coherence, making them ideal models for studying quantum effects in living systems.
It sounds like fluorescent proteins might be biological quantum devices themselves. Finally, let's touch on neuronal ion channels. This is an area that has some rather bold claims associated with it, but also very grounded potential. What role could quantum coherence play in the brain?
It seems unlikely at first glance, but neuronal ion channels are crucial for nerve, and thereby brain, function. With pores only around 0.1 nanometers diameter, ions travel single file. This nanoscale suggests they might exhibit quantum phenomena. The key concept applied here is quantum coherence, or QC, the coexistence of different quasi-classical descriptions of the same physical system. While usually short-lived, calculations have identified ion channels as preferential sites for non-trivial quantum effects in the CNS, potentially necessary to explain fast-directed permeation.
So how might ions, usually thought of as classical particles, behave quantum mechanically in these channels?
One study showed that within the selectivity filter of an ion channel, a distance of 0.3 nanometers between ions allows them to be described in a state of quantum superposition, consistent with wave-like propagation. In this scenario, decoherence might actually promote the energetic transition of the ion between binding sites, guiding its transport. Simulations that oscillated between fully coherent and completely classical states best replicated experimental observations.
That's a fascinating thought that decoherence could be a directional force. And there's also the idea of quantum tunneling for ions.
Yes, theoretical investigations have looked into quantum tunneling of ions through closed channels. One study implementing the Goldman-Hodgkin-Kotz equation found significant probabilities for tunneling of ions like lithium and potassium. This has led to proposals for a quantum electrochemical equilibrium model for ion conduction, potentially explaining therapeutic effects of lithium in bipolar disorder or the ion dynamics of potassium in phantom limb pain. There are also ideas about quantum-confined superfluids adapting to model nerve signal transmission for near-instantaneous responses.
These are all highly speculative but incredibly intriguing ideas. It sounds like quantum biology is truly a frontier pushing the boundaries of what we understand about life itself.
It absolutely is. The molecular machinery of life definitely involves individual electrons or protons, which inherently involve quantum phenomena. The real challenge is establishing a definitive biological function for these quantum features in living cells that contributes to fitness. Many current experiments are still on in vitro systems, and the complexity of even simplified biological systems makes quantum modeling difficult. The future lies in bridging this gap, transferring measurable quantum biological systems into more tractable organisms like E. coli, and developing new generations of interdisciplinary scientists. The field needs to move beyond in vitro systems, which skeptics often dismiss, toward engineered systems where quantum events and macroscopic outcomes can be simultaneously detected.
The field of quantum biology is clearly growing and promises to revolutionize our understanding of life.
The room is cramped. It smells of stale tobacco, metallic dust, and the damp, permeating chill of an English winter. It is late February 1953 inside the Cavendish Laboratory at the University of Cambridge. Two men are hunched over a desk that is less a workspace and more a battlefield of cardboard cutouts and metal retort stands. James Watson and Francis Crick are exhausted. They are trying to solve the structure of DNA, the molecule of life, but the pieces simply do not fit. They're sliding cardboard representations of the 4 DNA bases, adenine, thymine, guanine, and cytosine, across the table, trying to make them lock together like puzzle pieces. But in their current configuration, the chemistry is wrong. The hydrogen atoms are clashing. The bonds repel each other. The model is a disaster. It is a moment of profound scientific frustration. They are on the verge of losing the race to Linus Pauling across the Atlantic.
Then, a door opens. A visiting American chemist named Jerry Donahue walks in. He glances at their cardboard scraps and shakes his head. He tells them they are wasting their time. They are using the wrong shapes. You see, Watson and Crick were using what chemists call the enol and amino forms of the bases, structures where the hydrogen atoms are attached to oxygen or nitrogen. Donahue tells them that in the watery environment of the cell, these bases wouldn't look like that. They would exist in their keto and amino forms. The hydrogen atoms would be in different positions. Watson listens. He takes his cardboard scissors and trims the models to match Donahue's instructions. He shifts the position of the hydrogen atoms. He slides the new adenine toward the new thymine. Click. They fit perfectly. 2 hydrogen bonds form a seamless bridge. He tries guanine and cytosine. Click. 3 hydrogen bonds. The double helix is born in that quiet snap of cardboard. It is the most famous discovery in biological history.
But in the celebration that followed, in the rush to publish their famous paper in Nature, something was buried. A footnote, a whisper of a problem that wouldn't be fully heard for another 70 years. If Jerry Donahue was right, and the position of a single hydrogen atom, a single proton, determined how the code of life was read, what would happen if that proton moved? What if it didn't stay where it was supposed to be?
Today, we are going to descend into the subatomic basement of that 1953 discovery. We are going to look at the ghost in the machine of our genetics. We are going to explore how the strange, probabilistic laws of quantum mechanics, specifically a phenomenon called proton tunneling, might be the engine of spontaneous mutation. We are going to find out how a single proton teleporting through a solid energy barrier can rewrite the code of life. To understand this, we have to leave the macroscopic world of cardboard cutouts and descend into the violent, vibrating reality of a single DNA base pair. Imagine we are shrinking down, past the cell wall, into the nucleus, right up to the rungs of the double helix ladder. In the standard Watson-Crick model, the rungs of the ladder are held together by hydrogen bonds. A hydrogen bond is essentially a proton, a positively charged hydrogen nucleus, shared between 2 other atoms, usually a nitrogen on one side and an oxygen or nitrogen on the other. It's a game of tug-of-war. The proton sits in a comfortable valley of energy, closer to its parent atom, holding the 2 strands of DNA together.
Picture this landscape. You have 2 deep valleys separated by a high mountain ridge. The proton is a ball resting at the bottom of one valley. This is the canonical position. This is where the proton belongs to keep adenine behaving like adenine. To get to the other valley, to the other side of the DNA strand, the ball would need a massive kick of energy to roll up the mountain and down the other side. In the cold, hard logic of classical physics, the ball stays put. It doesn't have the energy to climb the ridge. The code is safe. The code is stable. But down here, at the scale of atoms, classical physics does not apply. We are in the quantum realm. And in the quantum realm, solid objects are not solid, and position is not a fixed coordinate. It is a probability cloud. This leads us to the phenomenon of quantum tunneling. In the quantum world, a particle, like a proton, behaves like a wave. And if a wave hits a wall, it doesn't just bounce off. A small part of the wave seeps through. If the wall, the energy barrier, is thin enough, there is a statistical probability that the particle will simply disappear from one side and instantly reappear on the other, without ever climbing over the top. It ghosts through the mountain. For decades, biologists dismissed this. They argued that the cellular environment is too warm, too wet, and too chaotic for delicate quantum effects to survive. They called it decoherence. They assumed the noise of life would drown out the quantum whisper. But recent research has shattered that assumption.
Let's jump forward to a different lab and a different era. It is 2020 at the University of Surrey. A team led by physicist Jim Al-Khalili and biologist John Joe McFadden, along with their doctoral student Louis Slocum, are staring at computer monitors. They aren't using cardboard cutouts. They are using advanced density functional theory and open quantum systems modeling to simulate the environment of a DNA strand. They are looking at that proton and the hydrogen bond. They want to know, does it tunnel? And does it matter? Their simulations revealed something startling. The proton doesn't just sit still. It is constantly flickering. It borrows energy from the warm, wet bath of the cell to tunnel back and forth across the energy barrier. It's a rapid, constant oscillation. Most of the time, it flickers over and flickers right back before anyone notices. It's a momentary glitch in the matrix.
But here is where the danger lies. This flickering creates a shapeshifter. When the proton tunnels to the wrong side of the bond, the DNA base changes its chemical personality. It undergoes what chemists call tautomerization. Let's visualize this transformation. Imagine an adenine base. It normally has a specific arrangement of protons that makes it want to pair with thymine. It has a specific shape, like a lock that only accepts the thymine key. But suddenly, a proton tunnels. It jumps from a nitrogen atom on the adenine to a nitrogen atom on the opposite thymine. Simultaneously, another proton tunnels back the other way to balance the charge. This is a double proton transfer. Now, the adenine is no longer standard adenine. It has become a tautomer, specifically the iminoform of adenine. It looks different. It acts different. Chemically, this tautomeric adenine now mimics the shape of guanine. It is wearing a disguise. Usually, this is harmless. The proton tunnels, the base puts on the disguise for a fraction of a nanosecond, the proton tunnels back, and the disguise comes off. The DNA returns to normal. But timing is everything. We need to set the scene for the catastrophe. It happens during DNA replication, the moment a cell divides. Imagine the replication machinery of the cell. It is a massive biological engine called the DNA polymerase. It moves along the DNA strand like a locomotive on a track, unzipping the double helix and building a new copy for the new cell. It is moving at incredible speed, reading the bases A, T, G, C, and grabbing the corresponding matching bases from the surrounding fluid to build the pair. The polymerase is a strict librarian. It works on shape and chemical bonding. It knows that A pairs with T. It knows that G pairs with C. It relies on the position of those protons to identify which base is which. Now, freeze the frame. The locomotive is thundering down the track. It approaches an adenine. But just as the polymerase jaws are about to clamp down to read the base, the protons tunnel. The adenine flickers into its tautomeric disguise. It effectively puts on a guanine mask. The polymerase doesn't know about quantum mechanics. It doesn't know about tunneling. It only sees the shape in front of it right now. It sees a base that looks, chemically, exactly like guanine. So what does it do? It grabs a cytosine, the partner for guanine, and snaps it into place. The locomotive moves on. The moment passes. The protons might even tunnel back to their original positions a split second later, but it is too late. The wrong base has been cemented into the new DNA strand. An adenine has been paired with a cytosine. This is a mismatch. When this new strand separates and replicates again in the next cell division, that cytosine will pair with a guanine. The original adenine-thymine pair has now been permanently replaced by a guanine-cytosine pair. The code has been rewritten. A mutation has occurred. And it happened because a subatomic particle walked through a wall.
This mechanism was actually hinted at by Watson and Crick in their 1953 paper, though they didn't invoke quantum mechanics explicitly. They just noted that the bases could exist in these tautomeric forms. It was the Swedish physicist Per-Olof Lodin who, in 1963, explicitly proposed the proton tunneling hypothesis. He suggested that the genetic code wasn't just written in matter, it was written in quantum probabilities. Lodin's idea was brilliant, but for 50 years it languished on the fringes. It was too hard to prove. Biology was wet and messy, quantum physics was cold and clean. The two disciplines rarely spoke to each other. But the work at the University of Surrey has changed the conversation. Their calculations show that this tunneling isn't just a freak occurrence. In fact, the local environment of the DNA, the specific way the water molecules cluster around the helix, actually lowers the energy barrier just enough to make tunneling surprisingly frequent. The noise of the cell doesn't destroy the quantum effect. In a strange way, it might actually facilitate it.
This is a profound realization. It suggests that the stability of our genetic code, the very thing that keeps us from dissolving into biological chaos, is balanced on a razor's edge of quantum probability. If the barrier were slightly lower, our DNA would be unstable, constantly mutating, unable to hold information. Life would be impossible. If the barrier were slightly higher, tunneling would never happen, and the mutations that drive evolution might slow to a crawl. Think about the implications of this. We tend to view mutations as errors, as mistakes, and often they are. They lead to genetic diseases. They're the primary driver of cancer. When we talk about cancer starting from a random mutation, we are often talking about this exact process, a polymerase misreading a shape-shifted base. But look at the other side of the coin. Without mutation, there is no variation. Without variation, there is no evolution. Without evolution, the primordial soup never produces a single-celled organism, let alone a human being capable of wondering about its own atoms. This means that quantum tunneling isn't just a bug in the system, it might be a feature. The fundamental fuzziness of the quantum world, the refusal of a proton to commit to a single location, provides the raw material for the diversity of life on Earth.
Let's go back to that room in Cambridge in 1953. Watson and Crick are trimming their cardboard. They are trying to make the world static, to make it fit into a rigid, understandable structure. They succeeded. They gave us the double helix, the icon of stability. But inside that structure, the ghost is still walking. The proton is still tunneling. Every time your cells divide, millions of times a day, there is a small, non-zero probability that a piece of you will disappear and reappear in the wrong place. It turns out that life is not made of rigid cardboard. It is made of probability. We are, quite literally, quantum creatures, surfing the wave function, surviving because, and only because, our atoms refuse to sit still. This story changes how we see ourselves. We are not just biological machines. We are the intersection of the macroscopic and the quantum, a place where the laws of physics that govern stars and the laws that govern atoms meet to create something new. The mutations that cause cancer and the mutations that drove our evolution from the sea to the land share the same root cause: a subatomic particle ignoring the rules of classical mechanics. The next time you look at a diagram of DNA, don't see it as a frozen staircase. See it as a vibrating, flickering, dynamic storm. And remember that the very reason you are here to see it at all is because sometimes the walls in that storm are just illusions.
I want you to picture a standard refrigerator magnet. You know the kind, the little souvenir you pick up at an airport. Maybe it's holding up a grocery list or a photo. It's not exactly a marvel of engineering. It's weak. But here is the thing that absolutely blows my mind. That cheap little magnet is roughly 100 times stronger than the magnetic field of the entire Earth.
It's true. Depending on where you are on the planet, the Earth's magnetic field is somewhere between 30 and 65 microtesla. It is incredibly faint. In terms of energy, it's arguably smaller than the thermal noise, the random heat vibration happening inside a single cell.
And yet, every year, a European robin can fly from Scandinavia to the Mediterranean, navigating thousands of miles, often at night, by locking onto this incredibly weak, barely there signal. If the signal's that weak, surely the noise of biology, the heat, the chemical chaos, should drown it out completely. It's like trying to hear a whisper in the front row of a heavy metal concert.
That is the perfect analogy, and for a long time, physicists and biologists assumed it was impossible. They thought, sure, birds migrate, but they can't possibly be using the magnetic field in a sophisticated way because the energy levels are just too low to trigger a chemical reaction. But they were wrong. The answer, as it turns out, isn't just biology, it's quantum mechanics.
We are talking about the radical pair model and a protein called cryptochrome. This is the stuff that sounds like science fiction, but it is actually happening in the eye of a bird. Today, we're delving into how life might actually be engineering quantum physics to survive. Alex, welcome.
It's great to be here, Chloe. This is one of my favorite topics because it forces us to rethink the boundaries between physics and biology.
So let's go back before we get to the quantum weirdness. How did we even figure out birds were using magnets? Because I assume for a long time people just thought they followed the sun or the stars.
They do use the sun and stars, absolutely, but in the 1960s, a German husband and wife team, Wolfgang and Roswitha Wilczko, did some famous experiments with European robins. They put these birds in funnel-shaped cages during migration season. Now, a migratory bird in a cage still wants to migrate. It will physically hop and scratch in the direction it wants to fly.
Right, the majestic migratory restlessness. I've heard of this.
Exactly. So the Wilczkos surrounded these cages with Helmholtz coils, essentially big copper wires that allowed them to manipulate the magnetic field inside the cage. They found that if they rotated the magnetic field, the birds rotated their hopping direction. If they nulled the field out, the birds got confused. This proved definitively that the birds had an internal compass.
But knowing they have a compass is different from knowing what the compass is made of. I think the common assumption is that they have little pieces of iron in their heads, right? Like a literal compass needle floating in their brain.
That was the leading theory for a long time, and there is truth to it. We know birds have magnetite, iron oxide crystals, often in their beaks, and for years scientists thought this was the answer. The magnetic field pulls on the iron pulls on a nerve, and the bird feels a tug. It's a mechanical sensor. But there was a problem with the magnetite theory when it came to the compass.
What was the problem?
The problem was light. Experiments showed that this magnetic compass only worked properly when the birds were exposed to blue or green light. If you put the bird in red light or total darkness, the compass stops working. Now, a chunk of iron in your beak shouldn't care what color the light is. Iron is iron. The magnetic field is there regardless of whether the lights are on.
So that suggests the sensor is photoreceptive. It's doing something with light.
Precisely. This led a physicist named Klaus Scholten back in 1978 to propose something incredibly radical. He suggested that the compass wasn't a physical object being pulled, but a chemical reaction being modulated. He proposed the radical pair mechanism.
And 1978 is pretty early for this, right? I mean, was he laughed out of the room? Because usually when you say quantum effects in biology, people get skeptical.
He was absolutely ahead of his time. You have to remember, quantum states, specifically the kind we're talking about, like entanglement, are incredibly fragile. Usually to maintain a quantum state, physicists need vacuum chambers and temperatures near absolute zero. Scholten was suggesting this was happening in a bird's eye, warm, wet, and noisy. It seemed physically impossible that a quantum state could last long enough to signal anything.
Okay, so let's brave the physics here. Break down the radical pair mechanism. What is actually happening in the bird's eye?
All right, so the hero of our story is a protein called cryptochrome. Specifically, in birds like the European robin, it's a version called cryptochrome 4, or Cry4. This protein is found in the photoreceptor cells of the retina. Now, cryptochrome has a special molecule bound to it called FAD, flavin adenine dinucleotide. When blue light enters the bird's eye, a photon hits this FAD molecule.
So, step one is just standard vision physics. Light hits a molecule, energy is absorbed.
Correct. But here is where it gets weird. That energy kicks an electron. This electron jumps from a neighboring part of the protein over to the FAD. This transfer creates what we call a radical pair. You now have two molecules, each with an unpaired electron. And because they were created simultaneously from the same event, these two electrons are quantum mechanically entangled.
Entangled, meaning their states are linked.
Yes, and this is where the spin comes in. Electrons have a property called spin. You can think of it loosely like a top spinning, although it's more complex mathematically. In this radical pair, the two electrons can either be spinning in opposite alignment, which we call a singlet state, or they can be spinning in the same alignment, which we call a triplet state.
Singlet and triplet. Okay, why does that matter to the bird?
Because the chemical products are different. If the pair stays in the singlet state, the electrons eventually jump back and the molecule resets. It's like nothing happened. But if they flip into the triplet state, the molecule changes shape or goes down a different chemical pathway. It produces a signaling chemical. So you have a fork in the road. Path A, singlet, or path B, triplet.
And I'm guessing the Earth's magnetic field is what decides which path is taken?
Exactly. And this is the magic. The radical pair is oscillating between singlet and triplet. It's flipping back and forth, extremely fast, millions of times a second. The Earth's magnetic field, weak as it is, exerts a tiny torque on the spins of these electrons. It changes the rhythm of that flipping. Depending on the angle of the bird's head relative to the magnetic field, the electrons will spend more time in the triplet state or more time in the singlet state.
Wait, I want to make sure I really get this. So, the magnetic field isn't pulling on the electron like a magnet pulls a paperclip. It's more like it's changing the probability of a chemical reaction?
That is the perfect way to put it. It's a chemical compass, not a magnetic one. Imagine a dance floor. You have pairs of dancers. These are the electrons. They can either dance close together, singlet, or spin apart, triplet. The Earth's magnetic field is like the music playing in the background. If the music is fast, more dancers end up spinning apart. If the music is slow, they stay close. The bird's brain just counts how many dancers are spinning apart.
That is fascinating. So, if the bird looks north, the music changes, the chemical reaction yield changes, and the neurons fire differently. But if this is happening in the eye, does this mean the bird literally sees the magnetic field?
That is the leading hypothesis, yes. It wouldn't look like lines on a map. Because this is happening in the retina, superimposed over their normal vision, it would likely manifest as a variation in brightness or color. We call it the heads-up display model. Imagine wearing augmented reality glasses. You look around the room and you see the world normally, but maybe looking north, there's a faint grayish shadow or a brighter patch floating in your vision.
So a robin flying at night might look toward the pole and see a brighter spot, and it just flies toward the light? That sounds almost spiritual.
It really does, and the specificity is incredible. A study published in Nature in 2021 by researchers at the University of Oxford and the University of Oldenburg really cemented this. They looked at cryptochrome 4 specifically. They compared the protein found in European robins, which are migratory, to the same protein found in chickens and pigeons, which are not migratory.
I love that science always comes back to chickens. What did they find?
They found that the robin's cryptochrome 4 was significantly more magnetically sensitive than the chickens. It seems that evolution has tuned the protein in migratory birds to be a better quantum sensor. It holds that quantum state just long enough to get a reading.
This brings me back to the skepticism you mentioned earlier. How is this possible in a warm, wet eye? You said quantum states are fragile. If I have a quantum computer, I need to cool it to near absolute zero so the heat doesn't scramble the data. A bird's eye is what, 40 degrees Celsius?
It's about 41 or 42 degrees Celsius in a bird. Yes, this is the central mystery of quantum biology. The key is speed. The radical pair reaction happens incredibly fast, on the order of microseconds. The theory is that the reaction happens so quickly that the thermal noise of the environment doesn't have time to destroy the quantum coherence. It's a race. The electron has to make its jump and feel the magnetic field before the heat of the bird's body scrambles the signal.
So they're surfing the chaos. They're using the speed of the reaction to beat the heat.
Exactly. And there's even some theoretical work suggesting that a little bit of noise might actually help the system, a phenomenon called stochastic resonance, though that's still being debated. But the fact remains, nature seems to have found a way to do quantum sensing at room temperature, something our best engineers struggled to do.
I want to touch on the controversy, or at least the nuance here. You mentioned the magnetite, the iron in the beak earlier. Is that theory dead, or do birds use both?
It is definitely not dead. The current consensus is shifting toward a dual sense theory. Think about how you navigate. You need a compass to know which way is north, but you also need a map to know where you are.
Right. A compass is useless if I don't know if I'm in New York or London.
Exactly. The radical pair mechanism in the eye, the cryptochrome, is almost certainly the compass. It tells the bird the direction of the magnetic field lines, but it can't tell the bird the intensity of the field, which varies across the globe and acts like a coordinate system. We think the magnetite in the beak might be the intensity sensor,
the map, so the bird sees the direction with its eyes and feels the location with its beak.
That is a sophisticated avionics package for a creature that weighs less than a battery.
It is humbling. And to add another layer of complexity, this compass is an inclination compass. It doesn't distinguish north from south like a Boy Scout compass. It distinguishes poleward from equatorial based on the angle the magnetic field lines dip into the Earth. If you took a robin to the southern hemisphere, it would still know how to fly toward the equator because it's measuring the dip of the lines, not the polarity.
So, if we accept that this is happening, that birds are exploiting electron spin dynamics, what does that mean for us? Does this change how we look at biology in general? Because for a long time, biology was just viewed as wet chemistry, balls and sticks sticking together.
It fundamentally changes the game. If natural selection can optimize a protein to exploit quantum entanglement, what else is it doing? There are theories now looking at olfaction, our sense of smell. There's a theory that we don't just smell the shape of molecules, but their vibrational modes, which is a quantum tunneling effect. There's research into photosynthesis, suggesting that plants use quantum coherence to transport energy with near 100% efficiency.
It makes life seem less like a machine and more like, well, like a quantum computer.
That's the frontier. The field of quantum biology is trying to prove that life isn't just living in a quantum world, but is actively harnessing it. The European robin is just the poster child because the evidence there is the strongest we have, but it opens the door to the idea that quantum phenomena are not just for physicists in labs, they are the machinery of life.
It's wild to think that this was all happening millions of years before we even discovered the electron. The birds figured it out first.
They absolutely did, and we are only just catching up. One of the most compelling pieces of evidence that really convinced the skeptics involved radio waves. You know how radio frequencies can interfere with electronics? Well, researchers found that if you expose birds to a very specific frequency of electromagnetic noise that is too weak to heat up tissue or trigger nerves, but exactly the right frequency to mess with the spin states of electrons, the birds lose their navigation.
Whoa, so they jammed the radar.
Essentially, this is called the Larmor frequency. By broadcasting a weak radio signal that resonated with the electron spin, they scrambled the conversation between the singlet and triplet states the birds couldn't orient. That was the smoking gun. A chunk of iron wouldn't care about that radio frequency. Only a radical pair would.
That is incredibly specific. It's like finding the exact frequency to shatter a wine glass, but instead you're shattering a bird's sense of direction.
And it brings up concerns about our modern world, too. We are filling the air with electromagnetic noise. While there's no evidence yet that 5G or Wi-Fi is causing mass extinctions of migratory birds, it is something researchers like Hendrik Mortson are looking at very closely. In fact, in some of those experiments, they had to take the birds out of the city and into the countryside because the urban electromagnetic noise was already interfering with the results.
That is a sobering thought. We might be blinding them without even knowing it. Before we wrap up, I want to go back to the visual. I'm trying to imagine being that robin. I'm flying over the ocean, it's pitch black, maybe just the stars above, and I have this glow in my vision. Do we know if it's in both eyes? Is it monocular?
That is a great question. Experiments suggest it is actually lateralized. For many birds, this magnetic sense seems to be located primarily in the right eye. If you cover the right eye, they get disoriented. If you cover the left, they are fine. This suggests the processing of this quantum signal is handled by a specific hemisphere of the brain, likely the left hemisphere, which processes input from the right eye.
So they are literally winking at the world to see where they are going. That is bizarre.
Biology is messy. It grabs whatever tools are available. If a protein that was originally designed for circadian rhythms, which is what cryptochrome usually does, it regulates the body clock, can be repurposed to detect magnetic fields, evolution will do it. It's the ultimate hacker.
Turns a clock into a compass. I love that. Alex, this has been a mind-bending tour of the avian eye. What is the one thing you want people to take away the next time they see a bird hopping around in their backyard?
I want them to realize that the bird isn't just looking for worms. It is interacting with the fundamental forces of the planet in a way we can't even feel. That little robin is performing a high-speed quantum physics experiment every time it opens its eyes.
We look at nature and think we understand it, but there are layers of complexity, quantum layers, that we are only just beginning to peel back.
A quantum physicist in feathers. Alex, thank you so much for breaking this down for us.
My pleasure. Thank you.
It really puts a new spin, pun intended, on the idea of a bird's eye view.
Picture a sidewalk in the middle of July. It's hot, maybe 80 or 90 degrees. The sun is beating down on the concrete, and right there, growing out of a crack, is a dandelion. It looks completely ordinary. It's floppy, it's green, it's probably covered in dust. But if you could shrink yourself down to the size of a photon, a single particle of light, and crash land onto one of those leaves, you would see something that shouldn't be possible. You wouldn't see a messy biological soup. You would see a machine operating with the level of efficiency that our best supercomputers and solar panels can only dream of. You'd see a system that seems to break the rules of classical physics, performing a magic trick that engineers have been trying to replicate for decades. We tend to think of quantum mechanics as this fragile thing that happens in sealed, sub-zero vacuum chambers. But today, we're finding out that the most sophisticated quantum laboratory might just be that weed in your driveway.
It really challenges the way we've traditionally drawn the lines between sciences. You have physics, which is precise and cold and mathematical, and then you have biology, which is warm and wet and messy. The idea that plants, these wet, messy things, could be harnessing the same quantum principles we use in high-tech computing, it sounds like science fiction, but the evidence is suggesting that life figured out quantum hacking billions of years before we even invented the calculator.
So that's where we are headed today. We are looking at the quantum leaf. I'm Jake, and I'm joined by Rachel to dissect how plants achieve near-perfect energy efficiency using quantum coherence. Rachel, before we get into the weird quantum stuff, we have to understand the problem the plant is actually trying to solve. Why does it need quantum mechanics? Why isn't regular old chemistry enough?
It comes down to speed and survival. When a photon of sunlight hits a leaf, it's captured by a light-harvesting antenna, specifically a chlorophyll molecule. That energy gets converted into something called an exciton. Think of an exciton as an energetic state, a baton in a relay race that needs to be passed through a forest of proteins to reach the reaction center. The reaction center is where the actual chemistry happens, where that light energy gets turned into chemical fuel.
Okay, so the photon hits the antenna, creates this exciton baton, and the baton has to get to the finish line.
Exactly. But here is the catch. The reaction center is actually quite far away in molecular terms. If that exciton baton took a random path, bumping into things like a pinball, it would lose its energy as heat long before it reached the destination. In the classical view of physics, we used to call this the drunkard's walk. The idea was that the energy just sort of hopped randomly from molecule to molecule until it lucked out and found the center.
The drunkard's walk, that implies a lot of stumbling and a lot of wasted time.
And wasted energy. If photosynthesis worked like a drunkard's walk, most of the solar energy would dissipate before it ever became food for the plant. But when we measure the efficiency of this specific transport step, getting the energy from the antenna to the reaction center, it's not 50% efficient or even 80%. It is often cited as being near perfect, sometimes upwards of 95 or even 99% quantum efficiency. The plant barely drops the baton.
Hold on, I want to clarify that number because I've heard people say solar panels are only 20% efficient. Are you saying a leaf is 99% efficient?
I need to make a distinction there. A leaf's overall ability to turn sunlight into biomass, like growing a new stem, is actually pretty low, around 1 to 3%. But the initial step, the quantum step, where the photon is captured and moved to the processing center, that is the part that is near perfect. No human-made wire or circuit moves energy that cleanly without loss.
Okay, so the drunkard's walk is out. The drunkard wouldn't make it 99% of the time. So how did scientists figure out what was actually happening?
The turning point came in 2007. A team led by Greg Engel published a paper in the journal Nature that really shook up the field. They were looking at the Fenner-Matthews-Olsen complex, or FMO complex.
The FMO complex. Sounds like an apartment building.
It's actually a protein structure found in green sulfur bacteria. These are bacteria that live deep in the ocean where light is incredibly scarce. They can't afford to waste a single photon. If they miss one, they die. Engel's team fired ultra-fast laser pulses at this complex and observed something bizarre. They saw quantum beats.
Beats? Like a rhythm?
Like an oscillation. Imagine you drop a pebble into a pond. You see ripples spreading out. If you drop two pebbles, the ripples interfere with each other. Peaks hit troughs, troughs hit peaks. That interference pattern is a signature of waves, not particles. Engel saw these electronic oscillations lasting for hundreds of femtoseconds.
Femtoseconds. That's a quadrillionth of a second. To me, that sounds like zero time.
To a human, it's nothing. To an electron, it's an eternity. The fact that these beats lasted that long suggested that the energy wasn't hopping like a particle. It was flowing like a wave. It was in a state of quantum superposition.
Superposition, that's the Schrodinger's cat thing, right? Being in two states at once?
Precisely. The standard analogy here is a maze. If you are a classical particle, the drunkard, you have to try every single path in the maze one by one to find the exit. You hit a dead end, you turn back, you try again, it's slow.
And by the time you find the exit, the energy is gone.
Right. But if you are a quantum wave, you don't pick one path. You flood the maze like water. You go down every single corridor simultaneously. The wave feels the entire structure of the maze at once. And because of that, it automatically locates the most efficient path to the exit and flows primarily in that direction. That is quantum coherence. The plant isn't guessing where the reaction center is. It's exploring every possible route at the same time.
That is mind-blowing. It's basically performing a parallel computation. It's a biological quantum computer solving the shortest path problem every time the sun shines.
That was the conclusion in 2007, and it launched this massive wave of excitement, pun intended. Everyone started looking for quantum biology everywhere.
But, and I know there is a but coming because you're grinning, this is where the physicist in me gets skeptical. You mentioned earlier that quantum states are fragile. We build quantum computers today, and we have to wrap them in gold, shield them from magnetic fields, and cool them down to near absolute zero. If a single air molecule hits the processor, the whole thing crashes. Decoherence. The wave collapses.
Right.
So how on earth does a dandelion sitting on a hot sidewalk, vibrating with heat, full of water molecules sloshing around, how does it maintain a quantum state? It should be impossible. It's too warm, too wet, and too noisy.
And that is exactly the question that sparked a decade-long war in the scientific literature. You had the quantum biology camp saying, look at the beats, it's quantum. And then you had the skeptics, people like Duane Miller from the Max Planck Institute. Around 2017, Miller and others revisited these experiments. They argued that what Engel saw wasn't necessarily electronic quantum coherence.
What did they think it was?
They argued it was just molecular vibrations. Imagine hitting a bell. It rings. That's a classical vibration. Miller's group suggested that the protein scaffold holding the chlorophyll was just shaking, and that looked like a quantum beat in the data, but it wasn't actually doing the fancy superposition magic. They basically said, biology is too messy for this. It's just shaking.
So was the 2007 paper wrong?
Not wrong, but perhaps incomplete. This is where the story gets even cooler. The consensus that has emerged in the last few years from researchers at places like UCL and the University of Chicago is that it's actually both. It's a phenomenon called vibronic coupling.
Vibronic coupling. Break that down.
It's the marriage of the electronic quantum state and the physical vibration of the molecule. Here's the twist. In a quantum computer, noise is the enemy. You want total silence. But in a plant, the noise, the heat, the vibration is actually required.
Wait, the noise helps? That's counterintuitive.
Think of it this way. Imagine you are trying to roll a golf ball across a very bumpy green. There are divots and hills. If you just tap the ball gently, that's the classical hopping, it might get stuck in a divot, a local energy trap. It doesn't have enough energy to get out.
Okay, so the ball is stuck.
Now imagine the ground is shaking, like an earthquake. If it shakes too hard, the ball flies off the green. That's too much heat. But if it shakes just right, the Goldilocks amount, the shaking knocks the ball out of the divot and allows it to keep rolling toward the hole. That is what the plant does. It uses the ambient heat and molecular vibrations to bridge the energy gaps that would otherwise trap the excitons.
So the plant isn't fighting the warm and wet environment, it's leveraging it.
Exactly. Pure quantum coherence would actually be too fast and too slippery. The energy might just oscillate back and forth forever without settling in the reaction center. The noise breaks the symmetry just enough to push the energy downhill to the destination. It's a collaboration between quantum mechanics and classical chaos.
That's a brilliant distinction. We humans build machines that try to dominate nature. We create vacuums. We freeze things to absolute zero. We try to force the universe to be perfect. Nature looks at the mess, the heat, the vibration, and says, I can use this to my advantage.
And that is why this research is so pivotal for future technology. If we can understand how the FMO complex does this, we can design solar cells that don't need to be perfect silicon crystals.
Right, because current solar panels degrade when they get hot. They lose efficiency. But a plant loves a certain amount of heat.
There is a group at the University of Chicago that's actually working on synthetic mimics of this. They're trying to engineer materials that support this vibronic coupling. The dream is a solar panel that works better on a scorching hot day in Arizona because it's using that thermal energy to assist the transport rather than fighting against it. We call it quantum heat engines.
Quantum heat engines. That sounds like something out of Star Trek.
It does, and it extends beyond just solar power. There are implications for sensing, too. If biology can detect single photons or weak magnetic fields using quantum effects, which birds likely do for navigation, by the way, then we could build sensors that are infinitely more sensitive than what we have now, operating at room temperature.
I want to go back to the controversy for a second because I think it highlights something important about how science works. You had Engel saying quantum and Miller saying classical vibration, and the answer turned out to be this nuanced middle ground. Is the debate settled now? Do we all agree on vibronic coupling?
Settled is a strong word in science. The debate has shifted. I think most people accept that there is a mix of electronic and vibrational states. The argument now is about how non-trivial the quantum part is. Is it just a byproduct, or did evolution actively select for it? Did plants evolve specifically to tune these vibrations?
That is a fascinating evolutionary question. Did natural selection act as a quantum engineer?
The evidence suggests yes. The spacing between the chlorophyll molecules in the FMO complex is incredibly precise. If you moved them just a few angstroms, a few atomic widths, the whole system would fail. The vibrations wouldn't match the energy gaps, and the exciton would get stuck. It implies that over billions of years, nature tweaked the geometry of these proteins until they hit that resonance frequency.
So the dandelion in the crack of the sidewalk is, structurally, a tuned instrument.
It's a Stradivarius violin playing a quantum symphony.
I love that image, but it makes me wonder, if this is so efficient, why haven't we seen it in other biological systems, or have we?
We are starting to. I mentioned bird migration. Robins seem to use quantum entanglement in their eyes to see the Earth's magnetic field. There is research looking into enzyme catalysis, exploring if enzymes use quantum tunneling to move protons around. Even the sense of smell, the vibration theory of olfaction suggests our nose detects the quantum vibrations of molecules, not just their shape. Photosynthesis was just the first domino to fall, because it's relatively easy to study with lasers.
It forces us to redefine what life is. We used to think of life as a chemical process, but maybe life is fundamentally a quantum process that just happens to use chemistry as its building blocks.
That's the frontier. The field of quantum biology is basically trying to answer the question, does life exist on the razor's edge of the quantum world? And the answer increasingly seems to be yes.
So let's wrap this up with a takeaway for the listener who is just looking at their houseplant right now. What should they be thinking?
They should realize that the quiet static green thing in the corner is actually a roaring engine of quantum probability. It's taking the chaos of the universe, the random bombardment of light and heat, and using the most sophisticated physics imaginable to create order and fuel. It's not just soaking up sun, it's surfing the probability wave.
And it's doing it all without a cooling system or a gold-plated vacuum chamber. It's doing it right there in the dirt.
Right there in the dirt.
Picture a quantum computer.
You've probably seen the photos, these golden chandelier-like contraptions hanging in a room that looks like a villain's lair. They are incredibly fussy machines. They have to be kept at near absolute zero, colder than deep space, in a perfect vacuum if a single air molecule hits a qubit, or if the temperature rises by a fraction of a degree, the whole calculation collapses. Game over.
Right. We call that decoherence. It's the enemy of quantum mechanics. The environment is constantly trying to bully quantum systems into acting like classical objects.
Exactly. So here's the thing that keeps me up at night. If maintaining a quantum state requires this extreme level of isolation and cold, how on earth is a spinach leaf doing it in my backyard?
That is the question, isn't it? That is the paradox that launched an entire field.
Because a leaf is the opposite of a quantum lab. It's warm, it's wet, it's chaotic. It is, to use the physicist's term, a noisy environment. And yet, we now have evidence that life isn't just avoiding quantum mechanics, it might actually be using it. Welcome to the show, everyone. Today we are looking at the impossible physics of living things. And joining me to unpack this is Eloise. Eloise, are we saying that plants are quantum computers?
In a very specific, limited sense, yes. But they're doing something even stranger than our best quantum computers. They aren't fighting the noise of the environment. They're actually using the noise to help them calculate. It's a concept called warm quantum biology, and it completely upends what we thought we knew about the fragility of the quantum world.
Okay, so before we get to the how, which I know involves some mind-bending thermodynamics, let's establish the what. Where is this happening? Are we talking about photosynthesis?
Primarily, yes, that's the poster child. Specifically, we're looking at the very first split second of photosynthesis. So sunlight hits a leaf, a photon crashes into a pigment molecule. That energy creates an excitation, essentially an electron that's been kicked up a level. We call this an exciton.
And that exciton has to get somewhere, right? It's like a courier with a package.
Exactly. It has to travel from the antenna complex, which is where it was caught, to the reaction center, which is the battery charger, where that energy is actually converted into chemistry. Now, if you look at this classically, like high school physics, the exciton would bounce around randomly. It would be like a drunk person in a maze, bumping into walls, taking wrong turns, eventually stumbling into the exit.
And that would be slow, and you'd lose energy.
You'd lose a lot of energy. If photosynthesis worked that way, plants would be terribly inefficient. But here's the kicker. In many photosynthetic bacteria, that energy transfer happens with nearly 100% quantum efficiency. Almost zero energy is lost. The courier doesn't just find the exit, it finds the absolute shortest path instantly every single time.
And this is where the quantum weirdness comes in, the finding the path part.
Right. In 2007, a chemist named Greg Engel published a landmark paper in Nature. He was studying the FMO complex, that's a specific light-harvesting protein in green sulfur bacteria. He shot lasers at it and observed something that shouldn't have been there: quantum beats.
Beats? Like a musical beat?
Sort of. Imagine you strike a bell, it rings. That ringing is an oscillation. In quantum mechanics, if a particle is in a superposition, meaning it's in two states or two places at once, you see interference patterns that look like oscillations or beats. Engel saw these lasting for more than 600 femtoseconds.
Femtoseconds sound short. I'm guessing that's actually a long time in this world.
It's an eternity. In the quantum realm, decoherence usually happens in tens of femtoseconds in a warm environment. 600 was shocking. It suggested that the energy wasn't hopping from stone to stone. It was acting like a wave. It was effectively taking all possible paths to the reaction center simultaneously and then collapsing only onto the most efficient one.
So the drunk in the maze suddenly becomes water flooding the maze. The water goes down every corridor at once, hits the dead ends, but also finds the exit. And because it's a wave, the main flow just naturally goes to the exit.
That's the classic analogy. It's a quantum walk versus a random walk.
Okay, but here's where I get stuck. And this is the paradox part. Engel's early work was at 77 Kelvin, liquid nitrogen temperatures. But plants don't live in liquid nitrogen. They live in the sun. As soon as you heat that system up, the molecular vibrations, the thermal noise, should destroy that delicate wave. It should smash the water into droplets. How does the wave survive room temperature?
This is my favorite part of the story. For years, physicists thought, well, maybe the coherent survives despite the noise, like the plant is building a really good shield. But newer research, including work by Seth Lloyd at MIT, suggests something much more radical. The plant is using the noise. The noise is actually necessary.
Wait, the noise helps? That sounds like saying static helps you hear the radio better.
It's counterintuitive, I know. It's called environment-assisted quantum transport, or ENACT. Let me try an analogy. Imagine you are walking down a staircase in the dark. But it's a weird staircase. The steps aren't even. Some are huge drops, some are tiny.
Okay, sounds dangerous.
Very. In quantum mechanics, energy levels are like those steps. For an electron to move from one pigment to the next, it often needs to drop down in energy. But in the quantum world, you can't just step down unless there's something to absorb that exact difference in energy. You can't have leftover energy. If the step is too big and you don't have a way to dump the excess, you get stuck. You're frozen on the top step.
Because energy has to be conserved. It has to go somewhere.
Precisely. This is where the noise comes in. Thermal noise is essentially the protein scaffold shaking. It's vibrating. Those vibrations, we call them phonons, provide exactly the right amount of energy kicks to bridge the gap. It's like the earthquake shakes the stairs just enough to form a little ramp between the steps, allowing you to slide down.
So the shaking of the environment matches the energy gap the electron needs to cross.
Yes. It's a Goldilocks situation. If there's too little noise, the electron gets stuck, called Anderson localization. If there's too much noise, the quantum wave gets smashed, the Zeno effect. But biology has evolved to sit right in the middle. The protein scaffold isn't just a bucket holding the pigment, it's tuned. It shakes at exactly the right frequencies to facilitate the quantum walk.
That is wild. So evolution has actually tuned the messiness of the leaf to act as a quantum bridge.
Exactly. The messiness is a feature, not a bug.
Okay, that's energy transport, but I've read about another area where this warm quantum stuff is happening, and it's even weirder because it's not about plants, it's about animals, specifically birds.
Ah, the European robin, the star of quantum biology.
Right. We've known for a long time that birds migrate using the Earth's magnetic field, but the field is incredibly weak. It's weaker than a fridge magnet. How are they sensing it?
For a long time, we thought they had little chunks of magnetite, basically iron, in their beaks, acting like a compass needle. And some birds do have that, but it didn't explain all the data. It turns out the sensor is likely in their eye, and it relies on something called the radical pair mechanism.
Radical pair. That sounds like a surfing duo from the 90s.
It does, doesn't it? But chemically, a radical is just a molecule with an unpaired electron. Electrons hate being alone. They have a property called spin, and they usually pair up with an opposite spin partner, up and down, stable.
Okay, so how do we get a radical pair?
Light. A photon of blue light hits a protein in the bird's eye called cryptochrome 4. This energy knocks an electron over to a partner molecule. Now you have two molecules, each with a lonely unpaired electron. But here's the quantum magic. Those two electrons are entangled.
Entangled. Spooky action at a distance.
In this context, it means their spin states are correlated. They can be in a singlet state, spins opposite, or a triplet state, spins parallel. And they oscillate back and forth between these states millions of times a second.
And where does the magnetic field come in?
That's the key. This oscillation between singlet and triplet is incredibly sensitive to magnetic fields. Even a tiny field, like the Earth's, can shift the balance, making the electron spend more time in the triplet state versus a singlet state. And because different chemical reactions happen depending on which state they're in, this ratio effectively translates the magnetic field into chemical signals.
So the bird isn't feeling a magnetic pole, it's literally seeing chemistry?
We think so. The hypothesis is that the bird sees a visual overlay, maybe a shading of light and dark, superimposed on its vision, which tells it where north is. It's a quantum heads-up display.
That is mind-blowing. But again, Eloise, I have to play the skeptic. The eye is warm, it's wet. Entanglement is supposed to be incredibly fragile. How does the state last long enough for the bird to notice?
This is where the debate gets heated. In the cryptochrome experiments, the coherence, the time these electrons stay in that sensitive quantum dance, seems to be just microseconds, but that is long enough for the chemical reaction to occur. However, you're right to be skeptical. There is a camp of physicists who argue that we might be overinterpreting the data.
Let's talk about that, because science is never a clean narrative. What's the counter-argument? Is it possible this isn't quantum at all?
Well, nobody denies that atoms and molecules follow quantum laws. That's trivial quantum mechanics. Chemistry is quantum. The debate is about non-trivial quantum effects, specifically long-range coherence and entanglement. Some researchers, like Duane Miller from the University of Toronto, have revisited those photosynthesis experiments.
And what did they find?
They argue that the beats Greg Engel saw, the oscillations, might not be electronic superpositions. They might just be classical vibrations of the molecule. Imagine a bell ringing. That's a vibration, but it's not a quantum superposition. If the beats are just vibrations, then the quantum walk theory takes a hit.
So it might just be a really bouncy molecule, not a quantum computer.
Possibly. But even the skeptics admit that the efficiency is real. And the recent work on cryptochromes is harder to dismiss with classical physics because it relies so specifically on spin states, which are a purely quantum property. You can't really have a classical radical pair mechanism.
It feels like we are on the edge of a new discipline. If this holds up, if nature really is harnessing thermal noise to protect quantum states, what does that mean for us? Can we copy it?
That's the billion-dollar question. Right now, we build quantum computers by isolating them. We try to silence the world. Nature took the opposite approach. She tuned the noise. If we can learn how to build materials that use phonon-assisted transport like a leaf does, we could revolutionize solar panels.
Solar panels that work at 99% efficiency?
Or just solar cells that work effectively in low light or that can move energy over longer distances without loss. And for the bird compass, that's the blueprint for ultra-sensitive magnetic sensors. Imagine navigation systems that don't need satellites, just a tiny vial of protein.
It changes how you look at a houseplant, doesn't it? It's not just biology, it's engineering at a level we can barely comprehend.
It really does. It blurs the line between physics and biology. We used to think life was just stuff that happened on top of physics. Now we're realizing life is digging deep into the quantum toolbox.
And maybe the reason life exists at all is that it figured out how to surf the quantum wave before the wave crashed. Eloise, this has been a trip. Thank you for breaking it down.
My pleasure. Keep asking the weird questions.
Always. And for everyone listening, next time you see a robin or a leaf, give it a little nod of respect. It's doing quantum calculus while you're just struggling to park your car.
Welcome to a deep dive into an emerging and truly fascinating field, quantum biology. Our discussion today centers on a comprehensive review article titled Quantum Biology, an Update and Perspective, published in Quantum Reports in 2021. It's a collaborative effort from a large team of researchers with Youngchan Kim and John-Jo McFadden among the lead authors.
That's right, Jake. This paper is a fantastic summary of the latest progress in understanding how living systems might exploit quantum mechanics. Traditionally, biology was seen as classical, while physics and chemistry were rooted in quantum laws. But this review explores how quantum phenomena like coherence, tunneling, entanglement, and spin, which are usually thought to be confined to the atomic realm, could play a crucial, non-trivial role in biological processes. It's about how life harnesses these effects for evolutionary advantage.
And when you say non-trivial, you mean these aren't just background quantum effects present in all matter, but rather specific instances where biology has evolved to actively use them to achieve a particular function or efficiency. It's a big leap from just saying everything is quantum to life uses quantum mechanics. What are some of the key areas where this is being investigated?
Precisely. The review covers several areas. One of the most active is enzymatic catalyzed reactions, particularly hydrogen transfer. Enzymes, the cell's workhorses, accelerate reactions to an incredible degree by factors in excess of 10 to the 20th compared to uncatalyzed reactions. For decades, there's been debate about nuclear quantum tunneling in these reactions. Given the small mass and inherent positional uncertainty of a proton, some degree of H-tunneling is expected, even in uncatalyzed reactions. The question is, do enzymes leverage this?
So how do researchers measure something as elusive as quantum tunneling in a biological system?
A key tool is the kinetic isotope effect, or KIE. This compares the reaction rate of a lighter isotope, like hydrogen, to a heavier one, like deuterium. Because of their mass difference, their tunneling probabilities differ significantly. If the KIE is much larger than the classical theoretical limit of about 7 at room temperature, it strongly suggests H-tunneling is a predominant reaction pathway, as seen in some enzymes with KIEs as high as 81 for SLO1.
That's compelling evidence. But what about the role of the enzyme's structure and dynamics? Does the enzyme merely provide a scaffold, or does it actively participate in facilitating this tunneling?
That's where protein dynamics come in. The degree of tunneling is highly sensitive to the barrier. Experiments investigating the temperature and pressure dependence of KIEs show a wide range of responses. It's not just about overcoming a static barrier. Rapid donor-acceptor vibrations, often called promoting vibrations, can compress the barrier, increasing tunneling probability. These dynamics can be fine-tuned by evolution. For instance, morphine reductase and aromatic amine dehydrogenase respond differently to pressure, which has been modeled successfully by understanding specific atomistic changes.
And to better study these dynamic quantum effects, the paper mentions light-activated enzyme systems. What's the advantage there?
Most enzyme reactions are thermally activated, which limits how precisely you can study their rapid chemical steps. But with a light-activated enzyme like protochlorophyll-allyl oxidoreductase, or POR, you can trigger catalysis with a single pulse of light. This allows for studying reactions over a much wider range of temperatures and timescales, from femtoseconds to seconds. POR, which is involved in chlorophyll biosynthesis, exhibits 2 successive H-transfer reactions that both proceed by tunneling, coupled to these promoting vibrations. The light's role is proposed to overcome the barrier for highly unfavorable hydride transfer chemistry.
So it's a paradigm system for dissecting the interplay between quantum tunneling and protein dynamics. This leads us to another major area discussed in the review, photosynthesis. The efficiency of photosynthesis has long been admired, but the idea that it might involve quantum effects, particularly at physiological temperatures, still sounds quite extraordinary.
It does. While the overall process of converting sunlight to biomass is thermodynamically inefficient, the initial steps of light harvesting and energy transfer to the reaction center achieve near-unity quantum efficiency. This remarkable robustness, even in warm, noisy biological environments, led to the hypothesis that quantum mechanical effects are at play. Initially, energy transfer was explained by Förster resonance energy transfer, or FRET, which describes incoherent hops. But it became clear FRET alone wasn't enough for strongly coupled chromophores, and many systems occupy an intermediate coupling regime.
And the direct evidence for this came from new spectroscopic techniques, didn't it? I recall the 2007 paper by Engel et al. Being a landmark.
Absolutely. Two-dimensional electronic spectroscopy, or 2DES, allowed phase-sensitive investigations. Engel's team, studying the Fenner-Matthews-Olsen complex in green sulfur bacteria, observed oscillating cross peaks at cryogenic temperatures, which they attributed to electronic coherences between chromophores. These ultra-fast quantum beats suggested that excitation was delocalized, potentially enabling a quantum walk to find the most efficient path to the reaction center. This was highly contentious, with critics arguing the oscillations were vibrational, not electronic.
So how did the field move past that criticism to demonstrate long-lived coherence at physiological temperatures?
Further studies using 2DES, some at room temperature, revealed quantum beats persisting for hundreds of femtoseconds, long enough to play a non-trivial role in energy transfer. A crucial concept that emerged is that the environment, far from destroying coherence, might actually enhance it. Theoretical studies predicted that intermediate noise levels, characteristic of room temperature, could optimize energy transport, a phenomenon sometimes called a quantum phonon antennae. Vibronic transport has also been shown to increase energy transfer by 10% in the LHC of algae, Chromonus mesostigmatica.
That's quite a counterintuitive idea, noise helping preserve quantum effects. Beyond photosynthesis, the review moves into magnetic field effects on spin-dependent reactions, often associated with magnetoreception in animals. This seems to be another highly debated area.
Indeed, radicals, molecules with unpaired electron spins, are common in biology. If these spins become correlated and their chemistry is spin-dependent, magnetic fields can influence their reactions. The most prominent example is the radical pair model for animal magnetoreception, such as in migratory birds. For a magnetic field effect, or MFE, to manifest, several stringent conditions must be met, like the spin correlation persisting for at least 1 microsecond for sensitivity to weak geomagnetic fields.
My immediate thought is the thermodynamic barrier. How can weak magnetic fields, like Earth's, which are orders of magnitude weaker than thermal energy, possibly influence biochemical reactions?
That's a critical point. The key is that the radical pair, at least transiently, is far from thermodynamic equilibrium. When a radical pair is generated simultaneously, the unpaired electron spins are initially correlated, often in a singlet or triplet state. These states have different reactivities. The magnetic field then has a window of opportunity to interconvert these spin states before they relax to thermal equilibrium. This interconversion is driven by local magnetic interactions of nuclear spins within the radicals, known as hyperfine couplings, and this process is magnetic in origin.
So it's not about changing the overall energy of the system, but rather subtly influencing the spin dynamics of transient species. What else is required for a biologically relevant MFE?
The spin correlation must persist long enough, ideally for about 1 microsecond, for sensitivity to weak geomagnetic fields. The radicals also need to be separated by a certain distance, typically around 3.5 nanometers, for the exchange interaction between their spins not to dominate and suppress interconversion. Finally, the reaction itself must be spin selective, meaning the outcome depends on the spin state. If the radical pair reacts too quickly before spin evolution can occur, or if other spin-independent reactions outcompete the spin-selective ones, no MFE will be observed. In the case of cryptochrome, questions remain about the exact identity of the magnetically sensitive radical pair, how it overcomes interradical interactions, and how it responds to extremely weak radiofrequency fields that imply unusually long coherence lifetimes.
Moving from macro-scale navigation to the microscale, the paper also discusses proton tunneling in DNA, which directly relates to the fidelity of genetic information and mutations.
This is a profound area. Watson and Crick themselves, in their 1953 paper, hypothesized that spontaneous mutations could arise if DNA bases adopted rare, less favorable tautomeric forms, leading to mispairing. In 1963, Per-Olof Loden expanded on this, suggesting that proton tunneling across the hydrogen bonds within a base pair could facilitate this tautomerization. If these tautomeric forms persist during DNA replication, they could lead to errors. This geometry mimicry can evade cellular fidelity checkpoints.
So is there experimental evidence that proton tunneling actually leads to these tautomeric mismatches in DNA replication?
Recent experimental evidence like high-resolution X-ray crystallography and NMR relaxation dispersion has confirmed the existence of Watson and Crick-like mismatches involving tautomeric bases in DNA and RNA duplexes. Computational studies using quantum mechanical molecular mechanical approaches are modeling the energy landscape of these proton transfers. A recent study observed a kinetic isotope effect in G-bond-T mismatches, suggesting proton transfer is involved, but further temperature-dependent studies are needed to confirm tunneling.
It's a complex interplay between quantum events and the broader cellular environment. The review then shifts to fluorescent proteins, or FPs, as a novel model system for quantum biology.
FPs like GFP have been invaluable in cell biology, but there's growing evidence of their anomalous photophysical properties. Traditionally, Fret theory assumed incoherent energy transfer due to the limited proximity of fluorophores within the FP's beta barrel structure and rapid vibrational decoherence. However, experiments have shown that FPs exhibit coherent dynamics at room temperature, which challenges classical understanding. For instance, EGFP showed a high coincidence to accidental ratio for photon pair generation, and Venus A206 dimers showed Davidov splitting.
What kind of evidence are we talking about for coherence in FPs?
In 2001, Sinelli et al. Reported surprisingly slow dephasing times in GFP,
Later, Young et al. Observed ultrafast energy transfer in YFP dimers, consistent with coherent energy transfer. More recently, studies have demonstrated the generation of polarization-entangled photons in enhanced GFP and excitonic coupling in dimeric Venus FPs at room temperature. These findings suggest FPs may have evolved mechanisms to prolong quantum coherence, perhaps due to the protective environment of their beta barrel structure.
That's a remarkable thought, that FPs might act as miniature quantum systems in cells. Finally, the review touches on quantum coherence in neuronal ion channels. This realm seems to border on the more speculative, given some of the theories out there.
Yes, while some proposals like the Penrose-Hameroff ORC-OR model for consciousness are highly controversial, research into ion channels themselves is more grounded. Neuronal ion channels are incredibly small, about 0.1 nanometers in diameter, which is a scale where quantum phenomena could conceivably play a role. They also exhibit exceptional ion selectivity and throughput rates, like the KCSA channels 10 to the 8th ions per second, that are hard to explain with classical mechanisms.
So how could quantum mechanics contribute to their function?
The idea is that quantum coherence could enable ions to exist in a state of superposition, effectively spreading their wave function across the channel's electric landscape, with decoherence enforcing directionality. Theoretical models incorporating quantum interference terms have replicated the fast onset of nerve firing. There's also theoretical investigation into quantum tunneling of ions through closed channels, with implications for understanding the therapeutic effects of lithium in bipolar disorder or phantom limb pain.
It seems clear that quantum biology is a rapidly expanding interdisciplinary field. From enzymes to photosynthesis, magnetoreception, DNA, and even neuronal function, researchers are finding intriguing hints of quantum effects. But what's the biggest challenge moving forward?
The major hurdle remains proving that these quantum phenomena play a functional role in living cells and contribute to biological fitness. Most experimental data comes from in vitro systems, which are simpler. The challenge is to bridge the gap to in vivo systems, possibly by transferring measurable quantum biological systems into more tractable organisms like E. coli, and using biomimetic systems for deeper understanding. It requires a new generation of scientists who can truly integrate physics, chemistry, and biology, much like biochemistry required merging chemistry and biology a century ago.